What I wanted to bring up last class is the organization of some of those finer structures that we talked about, and we've seen this already. The ventricles as they appear within the brain, and the relationship is what I wanted to look at. So for instance, we talked about the thalamus. Here you can see the thalamus, one of the major parts of the diencephalon. It surrounds the third ventricle. So if I fade this, maybe I'll just hide it. You can see how it sits right abutting the third ventricle. It surrounds the third ventricle, and the hypothalamus, I think we want right. The hypothalamus, this green shaded area, is exposed to the fluid in the ventricle at that point. That's where the circumventricular organs lie. So it has direct access in the wall, essentially, of the ventricle. And we can see our other characteristic structures, the pons, which is obvious, the midbrain, the pituitary gland. We also started discussing the basal nuclei. The old name for them is the basal ganglia. So let's add the basal ganglia in there. Remember, we had a couple different layers. The lens on the most lateral portion, the putamen, and then the globus pallidus. There's two portions of it, the medial and the lateral aspect. But see how they are so intimately related, one to the thalamus, and then sort of encased by the border of the ventricles. The caudate nucleus up here. These were really important in fine motor control. What we didn't get to see were the choroid plexuses. The computer app wasn't as comprehensive as this one. So you can see them in here. If I fade the ventricles, the long strands on the inside of the ventricles, those purpley red shadowy things inside, are the choroid plexuses. Notice how long they are. They extend all the way through the ventricles to always express and produce cerebrospinal fluid. All the way through the superior border of the third ventricle too. Down the cerebral aqueduct and then into the fourth ventricle as well. So this really, I think, helps bring all of these structures together that we've talked about. Hi. You need to come in? No? Okay. I actually didn't know that you could get in that way. Nice little secret escape. So all of those structures come together here. And I wonder if we can toss in the limbic system as well, because this is intimately related to these structures also. The limbic system we talked about is the emotional brain, where we'll see today the olfactory nerves end or deliver their signals, a large portion. All of these structures intimately and tightly related in the brainstem area, the lower order brain centers. Questions? Did that help at all? Put some things together? Okay. Learning's bred out of repetition, so we'll go back to that a fair bit. We left class, we had talked about the functional areas of the cerebrum. We introduced some of the main areas that we're going to discuss in a little bit more detail. And the first one that we had summarized was the primary somatosensory area. So this is what we left off with last class. The postcentral gyrus is where we find the primary somatosensory area. The ridge bordering the central sulcus on either side of the cerebrum. And then we mapped areas in the somatosensory cortex to the rest of the body. And this is where we get information about those areas, somatic information about those areas. Itches, tingles, vibrations, heat, etc. And the map along the surface of the brain was what we called the homunculus, the sensory homunculus, or little man, where the proportions are related to how much sensory information arrives from those respective areas. On the other side of the central gyrus is a very similar area, only it's not sensory, it's motor. The primary motor cortex on the precentral gyrus, the gyrus just anterior to the central sulcus. Here, similarly, there are points along the outer surface of the brain that relate to motor control of different areas in the body. Somatic motor control, voluntary muscle contraction of different areas in the body. And the distribution is similar. You see the motor homunculus here. Not the sensory homunculus, the motor homunculus. And it's very similar. The distribution, the relative proportions are similar. Larger areas are devoted to areas that execute more complex skills, complex movements. So large area devoted to the face and the hands. Hand dexterity, very complex. Facial muscles, very complex. Smiling, frowning, furrowing your brow when you don't understand what's happening. Where the sensory information, so this controls voluntary muscle activation, and again, larger areas for more complex movement. Well, the sensory information on the last slide related to the same side of the body. The map here controls the contralateral side of the body, muscles on the opposite side. And you remember way back when we introduced the medulla, there's 4 ridges on the front. We had the pyramids and the olives just lateral to the pyramids. And in very brief terms, we talked about the descending motor tracts crossing over, the decussation of pyramids. That's why motor nerves originating on one side of the brain execute actions on the other side of the body. The tracks cross over at the medulla. You can also externally stimulate this area, which is really cool. It's sort of a special interest thing, not a big part of our course, but transcranial magnetic stimulation, or TMS, is a way that you can activate these areas of the brain using magnets from the outside of the head to achieve a certain goal. And I got to do this when I was in grad school, not in undergrad. You start to walk, and TMS can make you turn or lean a certain way by activating different parts of the motor cortex. And you can see if there are any deficiencies in the relays as well. So it's a nice way to evaluate and diagnose some more complicated coordination issues. So 2 major areas, somatosensory cortex, motor cortex. A third that is not major, but I thought was kind of interesting, is Broca's speech area. And if I just bring the map back up to remind you where it was, it's this really small area here on the border of the temporal lobe, on the frontal lobe, at the border of the temporal lobe. Now this is shown here on the right-hand side, but for most individuals it's in the left frontal lobe, just for ease of the diagram conveying the information. The speech area is on the left-hand side. We'll talk about the right-hand side in a second. But the speech area, as you might expect, controls verbal actions, coordinates the pharynx, the larynx to help regulate breathing and speech at the same time. Not so much the tongue, that's a different nerve that we'll talk about today, but there is some input to the tongue. Helps coordinate muscles of breathing and of speech so that we don't You can execute both, not a problem for you there, but if I ran out of breath while I was speaking and couldn't coordinate breaths between my words, we might be in trouble. Speech, of course, much more complex, fine motor control of the muscles around the lips, and the tongue, and the cheeks, and breathing. I say simple, but it's really less complex. Oh, I didn't say simple. Good. It is less complex. Still complex. The transitions between inhale and exhale, activating the diaphragm, the intercostal muscles to raise the chest up while still speaking. It's a really coordinated interplay. So Broca's speech area largely executes the plan for language and makes sure that you can breathe at the same time. Knowledge of the language is not held here. Knowledge of the language is different. Knowledge of the language resides in Wernicke's language area. Again, left side of the brain, temporal and parietal lobes, near Broca's speech area. And it's near Broca's speech area because they are closely associated and perform very similar functions. You draw on the knowledge of a language here to execute speaking that language and conveying that information in Broca's speech area. It also allows you to decipher words that people say to you, to compare words that are coming in with the meanings that you have associated or stored already. And we don't often think about this. The formation of this area, for the most of us, has occurred in large part through development as we learn to speak our primary language, which I'm assuming is English for most of us. But this is likely where new connections are formed when you learn a new language. So you would modify the connections in this area when you learned a new language. And then the strength of those connections and how branched they are, how many connections there are, would be proportional to how fluent you are in that language. So, je peux parler français maintenant. Et peut-être tu me comprends. Right? For those of you that don't speak French, I said I can speak French right now and maybe you can understand me. So I've added another layer in this area. But while it sounds like I'm somewhat fluent, I'm really not. I'm not so fluent in French. But for something else, I just learned the other day that Brot is German for bread. So I have like one new synapse, one new connection for German in this area as well. Brot. Ich esse Brot. I eat bread. Not nearly as fluent. There's no connotation or intonation. I'm just relying on the connections I've made to know I eat bread. Those are words. As you develop language, the source information on the left side of the brain is paralleled by changes in the right side that convey emotion, that give tone, that interpret posture and inflection, and color the words that are being said. So I can pick up on sarcasm or rolling your eyes when we talk about this somewhat mundane topic, if it's in English. If it were in German, I'm so focused on the words because I don't have these reinforced connections, I don't pick up anything else. No inflection whatsoever. So I thought this was a really neat tangent, something that we all take for granted in large part. There are areas that are devoted in the brain to learning, storing, and interpreting information. And then these are connected with other areas. You know, a pencil is something. By a name, you give it a pencil, and you associate new words in different languages with that idea. So let's leave functional areas for now. There are a couple other major ones, and I gave a shout out to the prefrontal cortex. But these are ones that I wanted to introduce in a bit more detail. Before we round out the chapter, I also wanted to just take a moment to talk about brain waves. As you make a thought or formulate a thought, as you interpret sensory information, it's the net sum of the firing of neurons in those areas that allows us to make a memory. What is that? Interesting. It's the net sum of firing of neurons that allows you to see the color green, or to identify a chair, or to hear and remember the word Brot as German. Maybe that's the only thing you learned today, who knows? It's the firing of nerves, or neurons, that allows that to happen. And there's so much sensory information, neurons are always firing. And we can observe that firing in what we call brain waves. And there are a couple characteristic distinguishing brain waves. They're largely very busy, they're choppy, they're noisy, but there's a few different characteristics, and they're broken down into 4 categories here. And it's not so much that I care that you know the difference between alpha and beta and delta and theta waves, but this is super interesting, especially in recent years, as we've tried to decode brain waves. You can monitor brain waves from outside of the skull as well, just like you can control the primary motor cortex. It's one of those, it's like one of those caps you put on to dye your hair, where you pull hair through. You have one of those with sensors instead of hair poking out that allows you to sense brain waves in all the different regions of the brain. And where this is applied in its most cutting-edge form, I think, is in brain-computer interface. Have you heard of this term, brain-computer interface? No? Brain-computer interface is decoding brain waves and then using that map, that understanding, to control something from the computer's point of view. The idea being you can control prosthetic limbs by thinking about them, for instance. And some of the coolest research has been done at Duke University. The initial foray into this area was a monkey wearing this cap with access to a joystick, and it essentially played Pong. It had to move the joystick to move a cursor towards a target, and the target would move and the cursor would move too. And the researchers are trying to figure out, okay, what is the monkey doing to execute that action? Because they wanted to replicate it. If you know the signal, you can replicate the signal. So they monitor the monkey, and the monkey's sitting there, and it gets a reward. So every time it matches the cursor with the target, it gets a reward, and it does this. It's well trained to do that. So these researchers got an idea, okay, well, there are some defining features going on in this monkey's brain. We think we can control it. So instead of the joystick now controlling the cursor, without the monkey's knowledge, they started reading its brain waves and then moving the cursor according to the brain waves, not according to the joystick. So they can decode brain waves and then do something with them. And so the monkey's sitting there, and it's moving the joystick, and the cursor's moving like normal. And you'd think that's the end of the story, right? It's pretty impressive that you can control something by reading brain waves. You know what the coolest part is? The monkey stopped using the joystick. It sat there, and then it just thought about where the cursor should go, and it went there. Maybe it was lag in the processing of the information, but that is a phenomenal finding. Such an unexpected and really cool finding. All because we can read the activity going on in the brain. And this is only in its infancy. It's only going to advance. We're all going to be wearing exoskeletons pretty soon. Who knows, your Google Glasses could probably be controlled by your brainwaves as well. Pretty interesting stuff. I like those tangents. They're interesting. I'm not going to test you on them, but it's really cool to know what's going on in this area, I think. Let's summarize. We've covered a lot in this section. Functional, structural divisions of the nervous system. You've reinforced that learning in lab. You should know it by heart. We've covered already in depth protective layers for the central nervous system. The bone is obviously one of the biggest layers of protection. We'll talk about that when we get to the skeleton. Inside the bones of the skull and the bones of the spinal cord, we have the meninges and the cerebrospinal fluid that help float the central nervous system. The portions of the central nervous system that we discussed in this section are those of the brain. So we have the brain stem and the cerebrum. Lower areas and higher order areas of thinking. The brainstem had three main components, from the bottom or inferior to the superior components: medulla oblongata, pons, midbrain. You've seen those again in lab this week. Lastly, the cerebrum, with all of its defining features, gyri, sulci, fissures, has areas devoted to certain tasks: learning languages, controlling somatic actions, interpreting sensory information. And that sounds simple, but I assure you it is far more complex than we could ever imagine. This is where our understanding of the cerebrum is put on hold. I'm not going to say it stops. You'll continue to learn about this, I'm sure of it. But this is where we'll stop for now. Any questions before we move on? I'm looking down here this time because I missed you last time. No?